Today we'll be talking about the Krebs cycle. As you all could remember, we did previously glycolysis, where we use one molecule of glucose to form two molecules of pyruvate. Now the pyruvate that we have formed from glycolysis is going to be used to form acetyl-CoA, and that acetyl-CoA is going to be used in Krebs cycle. So let's do a rapid review on glycolysis. So as we discussed previously, we said glycolysis is a process that occurs where? In the cytoplasm, and it is used to form ATP. But finally, we are going to have two molecules of pyruvate. So the pyruvate that we have gotten from glycolysis, that is a 3-carbon compound, which is a keto acid. The pyruvate, we have formed two molecules of pyruvate, right? From glycolysis. So the two molecules of pyruvate that have been formed are going to be converted into a two, well, technically not two, but you can see that we're going to remove one carbon here, and we're going to have 2 plus coenzyme A. So we're going to form acetyl-CoA. So how is that going to happen? So the pyruvate is a keto acid, right? So it has a ketone group, C-double bonded O, and carboxylic acid group. So what's going to happen is we have to remove carbon dioxide. So 2 molecules of carbon dioxide, of course 2 molecules of acetyl-CoA are going to be formed. And where are we going to get the CoA from? Of course from coenzyme A. So why do we attach S to this? It's just to tell you that coenzyme A has sulfur in it. That's why you mostly see it written as. Some textbooks may not write the S there, and it's okay, but they just put the sulfur to tell you how important it is. So pyruvate is combined with this, so coenzyme A, and to form acetyl-CoA. What else is important in this reaction? Remember, carbon dioxide has been removed, right? But where is the hydrogen here? The 2 hydrogens. So we're having a hydrogen from here and a hydrogen from here. Where are they going to go? They are going to be combined with NAD+. So we are going to have NAD+. So let's assume we have 2 molecules of NAD+, nicotinamide adenine dinucleotide, and they are going to be converted into 2 molecules of a reduced form, NADH plus 2 protons. So this is what we're going to have. So we're having the coenzymes, NAD plus, coenzyme A. There are 2 of the coenzymes that are part of the enzyme. So the enzyme is called pyruvate dehydrogenase complex. So we're having pyruvate, and one of the main things that's going to happen is the removal. That's going to happen is going to be the removal of hydrogen, right? So the 2 hydrogens are going to be added to NAD+. So removal of hydrogen means dehydrogenation. So the enzyme complex is called pyruvate dehydrogenase complex. So mostly abbreviated as PDH. Pyruvate dehydrogenase complex. And the coenzymes are coenzyme A, NAD+, and also FAD is also one of the coenzymes. So this is not part of Krebs cycle. This is a transition. It is a transitional reaction which involves the conversion of pyruvate into acetyl-CoA. Now that we have acetyl-CoA, we can go to Krebs cycle. And again, it's very important to note that the 2 molecules of NADH that have been formed, what are we going to use it for? Use them for. First of all, we already explained how the 2 ways by which we get ATP, energy in the form of ATP, right? So we said we have substrate level phosphorylation and we have oxidative phosphorylation. Substrate level phosphorylation involves the formation of ATP directly without having to go through the electron transport chain. But in this case, NADH, 2 molecules of NADH that have been formed are going to go to the electron transport chain and form ATP. By oxidation, which involves the loss of the hydrogen or the protons, and phosphorylation, which is going to involve the addition of inorganic phosphates to ADP with the help of ATP synthase. So that's what we call oxidative phosphorylation. And approximately 5 molecules of ATP are going to be formed from 2 molecules of NADH. So we're going to have approximately 5 ATP molecules. So now that we have formed 2 molecules of acetyl-CoA, so acetyl-CoA. In reality, we have formed 2 molecules of acetyl-CoA from 1 molecule of glucose from glycolysis. So now that we have technically 2 molecules, if we are starting with 1 molecule of glucose, we are going to combine it with oxaloacetate. So one may ask, where's the oxaloacetate coming from? Well, you have to know that this one is a cycle. So this one is going to repeat itself. Okay? So the first reaction in the Krebs cycle, first of all, anyways, we'll talk about why is it called, well, Krebs cycle, and why another name is called tricarboxylic acid cycle, but we'll explain that better. So acetyl-CoA is going to combine with oxaloacetate. What is oxaloacetate? So it's a 4-carbon keto acid. So we're having what? 4 carbons, right? And we're having a ketone group and a carboxylic acid group. So this one is oxaloacetate. So we have acetyl-CoA and we have oxaloacetate here. What is going to happen in this reaction? So, well, most of you, sometimes you may need to know how the mechanism, how this is going to happen so that you can easily remember. But sometimes if you don't know how to do it, it's okay. So in this reaction, we're also going to add water to it. Water. So we're having 2 carbon compound, right? And we're having a 4-carbon compound. Why are we adding water to it? Remember, water is HOH, right? So what is going to happen is that the coenzyme A, the S-CoA, is going to combine with this hydrogen, and they are going to form coenzyme A, like HSCoA. That's going to be one of the products. And the OH will be attached to where the C double bonded O is. Next, what's going to happen? 1 hydrogen from here, from the CH3, is going to be added to the O, to the C double bonded O. So let's draw it like this. COOH, CH2, C, right? So 1 hydrogen here is going to be added to the O, and we're going to form OH, right? And next, what's going to happen? We draw the remaining. We're left with what? CH2, right? CH2, C double bonded O, then OH, like I already told you. This OH is going to be added to the C double bonded O to form OH, and the hydrogen here is going to be added to S to form coenzyme A. So now we're having how many carbons here? We're having 1, 2, 3, 4, 5, and 6, right? We're having 6 carbons, right? 6 carbons. How many carboxylic acids? We're having groups. We're having 1 here. This is 1 carboxylic acid group. We're also having another one here, and we're also having another one here. So we're having 3 carboxylic acid groups. So you can imagine why it is called TCA, tricarboxylic acid cycle, because you're going to have what? Citrate. This is what we call citrate. Compound is called citrate, and it's a tricarboxylic acid. So some may ask, how can I draw this structure in another way? For example, if you don't know this mechanism, first of all, you have to know that citrate is having 6 carbons and it's having 3 carboxylic acids. So, first of all, the name of the enzyme that forms this is called citrate synthase, because we are synthesizing citrate. So let me ask, how do you draw citrate in another way? Well, now you know that it is a 6 carbon compound, so you draw it like this. 1 carbon, 2nd carbon, 3rd, 4th, 5th, 6th. So you draw them this way. You draw the carbons this way. So 2, 2, 2, and you join them to each other. Now you already know that it is a tricarboxylic acid, right? So we are going to have 3 carboxylic acid groups here. COOH, COOH, COOH. Next, what do you do? At the center, that means on the 3rd carbon, for example, carbon number 1, 2, 3, you're going to have an OH group. Then you complete the structures. You already know carbon forms a maximum of 4 covalent bonds. So it means where you don't have the extra bonds, that means there should be hydrogen. H2 and H2. So this citrate is the same as this. So, but if you don't want to go through the mechanics of the reaction, how it goes, you can just memorize this, how to draw citrate. Next reaction. So now we have citrate here. We're going to convert the citrate into the next stage. Well, in some literature, there's one step that is skipped. So we're going to convert citrate to aconitate, and then mostly they put aconitate in bracket because it's like more or less unstable. It is easily converted to the next one, which we call isocitrate. So now we have citrate, right here. So I want to explain the mechanics of how we're going to convert citrate into isocitrate. So the first step that's going to happen is that we are going to remove water from citrate, meaning OH from one carbon and the other hydrogen from the other carbon. Remember, water is H2O, right? H2O. So, but there are 2 parts, right? So we have H and OH. So when you remove OH from this and remove H from the other, we form a double bond. So the rule is when you remove 2 groups from 2 neighboring carbon atoms, you are going to form a double bond. Meaning, we are going to form this. Remember, this and this are the same, right? So, let me use this to better illustrate. So now we're going to have a double bond formed. So this is like an intermediate. So we have removed water, right? Minus H2O. Now we have formed aconitate. Okay, so here we have aconitate with a double bond. Next, what we are going to do is to convert the aconitate into isocitrate. How is that going to happen? Now we're going to re-add. We're going to add water now. So we have removed water and we're trying to add water. Why are we doing this? We're doing it so that we can shift the position of the OH. We want to shift the OH from the 3rd carbon to the 2nd carbon technically. Because when you have the OH here, we want to count it technically this way. So we want to shift it from this number 3 to number 4 here. So what will we have? We'll have this CH2 COOH So look at this structure here. Remember, water is HOH. So in this context, what are we going to do? We're going to add the hydrogen where? To this carbon. The carbon with the less hydrogen, well, with no hydrogen. Then the OH will be added to this carbon. So here, we're going to have CH. Then here, we're going to have OH and COOH. Now, look at the difference between citrate and isocitrate. So citrate here, as you already know, both of them are tricarboxylic acids. Both of them have OH, one group of OH. Then both of them have 6 carbons. And the difference here is the position of the OH. So that's what we call isomerism. So that's even why we call it isocitrate. So it's an isomer of citrate. But what's the name of the enzyme that converts citrate into isocitrate, which is a 2-step process? The same enzyme. So the conversion of citrate to aconitate and from aconitate to isocitrate is called aconitase. Aconitase. Now we have isocitrate, right? So these are the first, 3 steps of Krebs cycle. Now that we have isocitrate, what's our next step? Is the conversion, Now that we have isocitrate, we need to convert it into something else. And what's that? There's a process which we call oxidative decarboxylation. Oxidative decarboxylation. What does that mean? As you already know, decarboxylation means to remove carbon dioxide. Oxidation, as in oxidative as in oxidation, means to remove hydrogen. So in this case, we are going to remove carbon dioxide. So now you have these options. So you have COOH here, COOH here, and another COOH here. So where are we going to remove our hydrogen, and where are we going to remove our carbon dioxide? So we're going to remove the carbon dioxide here, the carbon dioxide in the middle, from the carboxylic acid group in the middle. Then the hydrogen, we're going to convert the OH group into a ketone group. So meaning we're going to have a double bond. So now we're going to have CH2, COOH. So now we have C. So we're going to have CH2. Then we have C. Now we're going to have a double bond here. And then COOH. 1, 2, 3, 4, 5. So as you can see, what happened is that we removed carbon dioxide, and we also removed 2 hydrogen atoms here. And where are they going to go? We're going to add the 2 hydrogen to NAD plus to form NADH plus an extra proton. So let me ask, how many molecules of NADH? Well, it depends on how many molecules of glucose you began with. So if we started 1 molecule of glucose in glycolysis, that means we are going to form 2 molecules of pyruvate, 2 molecules of acetyl-CoA, 2 molecules of citrate, 2 molecules of isocitrate, 2 molecules of α-Ketoglutarate, and it means we are going to use 2 molecules of, for example, NAD plus to form 2 molecules of NADH. And you already know, 2 molecules of NADH are equivalent, well, it's equivalent to 5 molecules of ATP, approximately. Now, what have we formed? Remember, this is a crucial step, the conversion of isocitrate to α-Ketoglutarate. Why? Because isocitrate is a 6-carbon compound, and α-Ketoglutarate is a 5-carbon compound. The name of the enzyme that converts isocitrate into α-Ketoglutarate is called isocitrate dehydrogenase. You already know why, right? That we are removing 2 hydrogen atoms from isocitrate and adding them to NAD+ to form NADH and a proton. So the enzyme is called isocitrate dehydrogenase. Now, why are we calling this one α-Ketoglutarate? So that's also one of the tricks you have to know that you should know. For structures, for example, this compound is having 5 carbons, right? 1, 2, 3, 4, 5. And 2 carboxylic acid group is like what we call glutaric acid or glutarate. Alpha-keto. In IUPAC system, one want to name some compounds using the alpha beta system. We take the carboxylic acid group as the main functional group, and the first carbon after the carboxylic acid group is said to be the alpha carbon. The next one is said to be the beta carbon, and so on, gamma. Okay, so because the ketone group is on the first carbon after the carboxylic acid group, that's why I call it α-Ketoglutarate. Next reaction. Now that we have a 5-carbon compound, by a process known as oxidative, let me write it here, oxidative decarboxylation. Now we're going to do the same. We're also going to undergo oxidative decarboxylation of α-Ketoglutarate. But how is this going to happen? Of course, carbon dioxide is going to be removed. So we're going to remove the proximal carbon dioxide. From the proximal carboxylic acid group. So now we're going to have COOH, CH2, CH2, C double bonded O, right? So the carbon dioxide has been removed. Next is oxidative, right? So we are also going to remove the hydrogen and add it to NADH. But first of all, what other compound do you also need to know? That we are going to add coenzyme A to it. Coenzyme A. So meaning we're going to have 2 available hydrogen atoms. One H from here, another H from here. So we're going to add it to, so meaning NAD+ is required. So we are going to remove the hydrogen from here and the hydrogen from here and add it to NAD, and we're going to form NADH plus a proton. Then the S-CoA will be added to the carbon. Coenzyme A. As you already know, like I said before, that we're going to form 2 molecules of NADH because we are starting with 1 molecule of glucose, which formed 2 molecules of pyruvate and so on. What's the name of the enzyme? Just like here where we had isocitrate being converted into α-Ketoglutarate, given that hydrogen atoms were removed, we call the enzyme isocitrate dehydrogenase. The same thing is happening here, meaning α-Ketoglutarate dehydrogenase complex. A complex. So we are going to have a lot of coenzymes here. NAD plus, coenzyme A, even FAD. These are the coenzymes of the alpha-ketoglutarate dehydrogenase complex. Alpha-ketoglutarate dehydrogenase complex. So what are we going to form here? So since you're having coenzyme A, I remember we're having 4 carbon, right? So we're going to call it Succinyl-CoA. Succinyl-CoA. So given that it has coenzyme A to it, and if we're having a dicarboxylic acid that is having 4 carbons, is succinate. So in this situation, it is succinate with a coenzyme A. Then we'll call it Succinyl-CoA. And anyways, we'll also talk about the regulatory steps in Krebs cycle. We have 3 regulatory steps. The step catalyzed by citrate synthase, the one catalyzed by isocitrate dehydrogenase, and the one catalyzed by alpha-ketoglutarate dehydrogenase complex. Now that we have Succinyl-CoA, we need to convert Succinyl-CoA into succinate. So, you already know, right? Succinyl-CoA means succinate with coenzyme A. That means we have to remove the CoA component of it. And we're going to form So now we have a pure dicarboxylic acid where you're having 2 carboxylic acid groups and 4 carbons. This one is what's called succinate. I hope by now you already know the meaning. Succinate means succinic acid, just like glutamate, glutamic acid, right? Pyruvate, pyruvic acid. Citrate, citric acid. And so that means we are going to remove the coenzyme A. But one very important thing is going to happen here. What's going to happen? We are going to have GDP, an inorganic phosphate added to GDP to form GTP. So this one is an energy yielding reaction where energy in the form of GTP or slash ATP is going to be formed. The name of the enzyme is called Succinyl-CoA synthetase. Succinyl-CoA synthetase. Very important reaction to note. And the energy in the form of GTP that has been formed is by a process known as substrate level phosphorylation. All the other ones, NADH, will only get energy if we go through the electron transport chain. And that process is known as oxidative phosphorylation. Next, the succinate that we have gotten, or the succinic acid that we have gotten, now can be converted into subsequent products. So succinate, what I'm going to do here is to remove two hydrogen atoms from these 2 carbons. So we're going to have, like I said before, that when we remove 2 groups from 2 neighboring carbons, we form a double bond. So a double bond has been formed between these 2 carbon atoms, right? So now, instead of succinate, we have what's called fumarate. What is the name of the enzyme that converts succinate into fumarate? The enzyme is called succinate dehydrogenase. The coenzyme is called FAD. FAD is converted into FADH₂. So flavin adenine dinucleotide is going to be converted into a reduced form of that. How many molecules? Of course, expected 2 molecules of FAD should form 2 molecules of the reduced form of FAD. And for FAD, 1 molecule of FAD yields approximately 1.5 ATP molecules. So 2 FAD molecules should give us approximately 3 ATP molecules. Now that we have fumarate with a double bond, we are going to add water to it. Water is HOH, right? HOH. So we're going to add hydrogen to one of the carbons and OH to the other carbon. Here it doesn't matter where to add it, right? You can add it here because it's basically 4, right? When you add OH here, it's the same as adding OH here. Because at the end of the day, when you're counting, you count so that the OH gets a smaller number. Now we have C, so with an OH, OOH, like this, CH2 here, because we're adding hydrogen to the carbon here. So the compound is called malate. Malate. So the enzyme, given that we're adding water, the enzyme is a lyase. You know, the classes of enzymes, check our other video on the classes of enzymes. So lyases will break a double bond to add a group. Here is not a hydrolase. Why? Because we're adding water not to split the compound into 2 parts. We're adding water so that we can break the double bond and add the group OH. So it belongs to the class of lyases. The enzyme is called a fumarase. Fumarase. It is a lyase enzyme. It's not a hydrolase. So next, the malate that has been formed is going to be converted into what? A ketone. So we're going to have double bonded O, COOH, CH2, COOH. So we're going to remove 2 hydrogen atoms from malate. And these 2 hydrogen atoms are going to be added to NAD plus to form NADH and proton. So if you have 2 molecules of this, you form 2 molecules of H. Now what have we formed here is oxaloacetate. Oxaloacetate. So what do you need to know? Since we have removed hydrogen from malate to form oxaloacetate, we need an oxidoreductase, a dehydrogenase. So malate dehydrogenase, malate dehydrogenase is the enzyme MDH to form oxaloacetate. The formation of oxaloacetate technically marks the end of Krebs cycle. So the cycle is going to repeat. So when we're going to have oxaloacetate being combined with acetyl-CoA to form malate, citrate, then citrate to form isocitrate, α-Ketoglutarate, Succinyl-CoA, Succinate, Fumarate, Malate, and the cycle repeats.